Thank you, Guillermo, very much for inviting me to be here. I feel I'm here under somewhat false pretenses because all of you are going to be discussing at this meeting how development works and how different kinds of cells are formed, whereas I'm going to be talking about when all that's happened, what is it that stops them going back again? What stabilizes the state that they've arrived in? Now, we're familiar with the normal pattern of development, so that as the egg forms the embryo, these various cell types are formed, and we know that there is really no normal reversal of this, so you wouldn't get peripheral nerve turning into heart or muscle into intestine. It's essentially a one-way process, sometimes amplification along the way, but under normal circumstances, cells never go backwards in development. So it's a very stable process of differentiation. But we might ask, why do we say that? How sure are we that that is really true? And the best test for this is to transplant tissue from its normal position into another environment so that it's surrounded by all sorts of different cells, and ask if the cells will now change to be in accord with the new environment. But this is not an entirely satisfactory test because when you move a piece of tissue, you normally move hundreds or even thousands of cells, and any one cell will, in general, be surrounded by its previous neighbors. So the test one really wants is one where you move one single cell into a new environment. And this experiment does that by taking a cell from this region of an embryo, and this is a cell which is going to be a future muscle cell, so it's a muscle progenitor, and it's transplanted, a single cell transplanted into the future gut region, endoderm, of an embryo. The cell may divide or not, but in the course of time, that cell, if it's divided, there are two of them, end up in the endoderm, in the intestine region of the embryo. And in cases like this cell is still a progenitor. It has not yet become a muscle cell, and it does so during its residence in the endoderm. But when it arrives in the endoderm, it then actually turns on its muscle genes and becomes a muscle cell. So in this case, this cell has undergone its intended differentiation into muscle in completely the wrong environment, surrounded by cells of an entirely different kind. So that says that cell is completely determined or committed in its development to form muscle. Doesn't matter what other cells are around it. And so in that sense, we can reasonably say that there is a great stability of that pathway of development. Now that applies to the whole cell. These are whole cell transplants. And this just shows you there is a muscle cell expressing muscle genes in the endoderm, just like the normal muscle cells do. Now, this stable cell differentiation can be reversed by somatic cell nuclear transfer or by induced pluripotency, by overexpressing transcription factors. And the major question now is what is the mechanism of this reversal? And I will be talking about the nuclear transfer aspect of that, largely because the process is relatively efficient, and so it becomes feasible to try and analyze the mechanism of this reversal of the stable state. And what I mean by nuclear reprogramming is the resetting of gene expression back to, from an adult back to an embryonic state. So the basic design of experiment, old-fashioned one, is to transplant a nucleus to an egg from which it may be possible to obtain a frog, or likewise in mammals. So this nucleus, importantly, is no longer surrounded by its own cytoplasm, but it's now surrounded by a new kind of cytoplasm, and that is sufficient to generate this change or instability compared to what that nucleus would have done if it had been left in its own normal cytoplasmic environment. So there are, in effect, two questions which one attempts to answer, and I shall deal with these in turn. One is, what are these components of eggs and embryos that can reprogram the somatic nucleus or reverse its stability? And secondly, what causes the resistance of somatic cell nuclei to these reprogramming factors? And that, of course, reflects very much the stability of the differentiated state. So just a little bit of background before we get underway. And this is an example of the reprogramming. So you have here the intestine of a feeding larva with its food inside, and that can be, nuclei can be transplanted, and as a matter of fact, this is a GFP-marked intestine. So when you do a nuclear transplant like I've just mentioned, you can obtain a larval tail with these single muscle cells, GFP-marked because they derive from intestine, and they are nicely integrated with the muscle of the host, normal tadpole, which are, of course, not GFP marked and are shown there. So there's this fairly dramatic switch from the intestine to muscle, and this is the kind of reversal which we're talking about. So we need to say just how efficiently does this process work. The first nuclear transfers of the kind that I've talked about will yield about 15% of cases in which you can go from intestine to functional muscle and nerve. That's the success of that. You can do serial nuclear transfer, where you take the first transfer embryos, take cells from those, and then retransplant the nucleus again to another set of eggs, and that can give a further addition to the efficiency of about 7%. And then you can graft from the first transfer embryos into hosts. And in these cases, these first transfer embryos are not normal and so would not develop. But if you do the serial transfer or the grafts, then you can get this switch of cell type, ending up with roughly a 30% success rate of this switching, of this reprogramming. So now we come to the first question, and that is, what are these components of eggs and oocytes that do the reprogramming? Now, the first point to make is that when you transplant a nucleus from a somatic cell, it has a very slow DNA replication rate. And after transfer, it does not, often does not accelerate sufficiently to become in accord with that of the egg, which will undergo its own replication rate. And this causes a lot of abnormality. So I'll try and intersperse a few generalizations in this talk, and one of the questions which people are often interested in is whether DNA replication and cell division is either necessary or helpful for resetting the nuclear reprogramming. I will argue that it is not necessary, though it can help. Now, the cases where it's clearly not necessary are a few here. One is when you transplant nuclei to oocytes, which we'll talk about that later. It's a very simple situation where you reprogram the nucleus completely in gene expression, but there is no DNA synthesis or cell division at all. Then in the mouse germline, as Msirani has shown that you can get nuclear reprogramming in the female germline, again without any cell division or replication. In these cell hybrid experiments, cell fusions, again, you can get major switches in the absence of replication or cell division, and a rather spectacular experiment by Melton was to switch exocrine to endocrine pancreas by the Yamanaka factor route or pancreas transcription factors. And again, you do not need cell division. So I think it's clear that cell division and replication are not necessary for completely resetting the expression pattern of a nucleus. I put at the bottom, but it can help, and that's because there are some experiments where, although there's not a necessity for DNA synthesis or replication, it does seem to help a little bit. So that's a generalization on this subject of whether replication and cell division are needed for this switching of gene expression. Now, the experiment that I've discussed so far is essentially this one, where you transplant a nucleus to an egg. That is followed by several rounds of DNA replication to reach the embryo stage. At this point, there is no transcription, but replication errors occur at this point, and that makes it very difficult to analyze the mechanism of the transcriptional switching, because it's confused by errors in DNA replication, and in any case, it doesn't even show up until a later stage. So one way of overcoming this is to essentially simplify the process, make the process much simpler to analyze, and one can do this by making use of these so-called first meiotic prophase oocytes. And the diagram of this works this way. Here is what I call an oocyte, and actually that is it's an egg progenitor, the growing egg cell in the ovary, and it's in the first meiotic prophase. It has a very specialized nucleus called the germinal vesicle, and you can inject multiple nuclei into that germinal vesicle. These transplanted nuclei undergo intense transcription, but no DNA replication, and the same nuclei will then show new gene expression, will be changed in gene expression as a result of residence in this germinal vesicle, which therefore contains a high concentration of reprogramming components. And this is what the first meiotic prophase oocyte looks like, a large cell, and that is the germinal vesicle, which cannot be seen unless you take it out, but you can take it out, and that's what it looks like. And that germinal vesicle is packed full of a huge abundance of components which normally will be transferred to the egg, second meiotic metaphase cell, during this normal maturation process, and these components will then appear or persist in the embryo, and they are necessary for normal development to take place. So you have here a kind of package of developmentally important components which are able to directly reset or reprogram somatic nucleus to a new pattern of gene expression. And when you analyze the transcriptional pattern of these such transplanted nuclei, you find that they turn on the pluripotency genes, well-known ones, often rather fast. This is within a day at, say, 17 degrees centigrade, which is really very fast at that temperature. And interestingly, if you use the nuclei of specialized cells, they take considerably longer, and they show this resistance to being reprogrammed, even though they're put in exactly the same environment of the germinal vesicle. One can follow the polymerase activity in these transplanted nuclei, the various forms of polymerase, including the elongating form, and essentially all of the nuclei become fully loaded with the polymerase. And the other really important, crucial thing about these sorts of experiments is the reinitiation of transcription. And you may know that if you were to make an extract of a cell and put isolated nuclei into that extract, they will not reinitiate transcription. Polymerase may run off, but it can't start again. Whereas if you transplant such isolated nuclei into the living cell, as in this case, you can get extensive reinitiation of transcription. So for SOX2 gene, it's about 600 transcripts per gene per day. We measure the absolute numbers in these cases, and so you have a system where the polymerase is reinitiating continually after the transfer, and therefore it's a very good system to analyze what is regulating this transcriptional activity. So the ability to test reinitiating transcription from a suspension of isolated nuclei is really crucial for these kinds of experiments. Now, it's helpful to put this whole process in the context of development, and my suggestion is that the way development actually works is to reprogram a nucleus during the oogenesis, the oocyte growth period, and this would, for example, apply to the nucleus that is in the oocyte. It has to be reset back to the origin, the original state of an unspecialized state. And in real life, the oocyte nucleus undergoes these processes during the rather long process of the oocyte growth and germ cell formation. When that's all finished, the oocyte will undergo hormonal maturation to form the so-called egg. The egg then divides, and eventually you get the embryo cells departing in different directions of cell differentiation. So sperm would normally enter at this point and would need to be reprogrammed before it can enter the further steps of cell differentiation, and the oocyte nucleus is already being reprogrammed at that point. So the idea here is that we need to know two things. One is, how does a specialized nucleus go back in development to become embryonic? And later one will want to know how that, having become embryonic, how it then starts to go through the normal differentiation processes. And my view would be that if we could understand how to get to this stage where the differentiated state has been fully reversed completely, then it would be the normal processes of development, a lot of signaling and so on, which we'll talk about at this meeting, which will apply to any nucleus which has been fully reversed in its differentiation. So I'm going to be concentrating on this part, on how you get a specialized nucleus reprogrammed back to the beginning again so that it can be reset to follow the normal steps of development. And there are 3 steps, which I've mentioned. Now this one, I shall now start by talking about the first step, which is called decondensation. And this slide from Magdalena Zernicke-Goetz shows sperm, highly condensed nucleus, and after fertilization, this nucleus swells up to an enormous structure with a massive decondensation of chromatin. And that's normal fertilization. When you transplant nuclei, we usually use a few hundred, into the oocyte, they also show this rather massive swelling and enlargement and chromatin decondensation of about 30-fold. And I might mention another generalization at this point. We actually use mammalian nuclei in the frog oocyte. And it turns out that in most experiments where people make these cross-species combinations, they work surprisingly well. So there seems to be rather little incompatibility between quite unrelated species in these regulatory events. It's very notable in cell fusion experiments and in nuclear transfer that the mechanisms seem to be insensitive to the differences between species. So one really important component of this whole process is what's called an oocyte-specific linker histone. And this was discovered not by some ingenious genetic screen, but simply by looking for the most abundant components in the oocyte. Now this linker histone, it's called B4 in amphibia or HF100 in mammals, similar thing. It's rather like the somatic linker histone, but it has a very different N-terminal end. And it is really oocyte specific. It's only really expressed in oocytes. Now, what you can do is to show that there is a remarkable replacement of the somatic linker histone by the oocyte-specific one. And in this experiment, we've started with some nuclei largely containing the somatic GFP-marked linker histone, and they've been transplanted to an oocyte which is supplied with the red fluorescent protein B4 histone. And we should see this in real time, this replacement. These are time-lapse pictures showing the nuclei. Of course, they move around a lot and mix up with the germplasm. I'll just do that one again. And you can see that, notably, nearly all of the nuclei are now fully enriched with the oocyte-specific linker histone. That one is interesting because that actually is a complete whole cell, not a nucleus. And it illustrates rather nicely the point that I started off at the beginning. If you deal with a whole cell, the nucleus is unaware of its environment, and that cell is very stable. But once you make nuclei, these ones, they will then sense their environment and be occupied by the molecules from the oocyte. And this process is really quite fast. It's largely happened in a couple of hours. This replacement, very soon after the nuclear transfer is this exchange taken place. A problem in this field is that of proving that any particular process is necessary rather than just a correlation. And we can't, it's no use doing gene knockout experiments because the protein's there. And it's no use doing message depletion experiments, again, because the protein is already there. So you have to do protein knockdown experiments. And this is done using either a dominant negative competitor or an antibody against B4. And you can see that with the control of C-Jun, which remains more or less constant, here is the normal switching of upregulation of pluripotency gene SOX in a day, and that upregulation is essentially eliminated by the B4 dominant negative or the antibody, arguing then that this is a necessary step for this reprogramming. In support of that, is the fact that FRAP experiments show a rapid exchange and loosening of chromatin. Rather surprisingly, you can take the germinal vesicle out of the oocyte and prepare it under glass slides, so you can carry out the FRAP experiment, and you can even do it by isolating the germinal vesicle and then injecting nuclei into it. The whole process of this reprogramming will take place in an isolated nucleus. Now, I have to say that is isolated in oil, so there's no aqueous exchange. But when you do that, you do see a significant change in the protein mobility in accord with this reprogramming and occupation of the nucleus by B4. Now, the next step in the process I will pass over fast. This is DNA demethylation of promoter sequences. That happens, but it's by no means true or needed for many genes. Many genes in specialized cells do not have methylated promoters, and although the oocyte can do it, in many cases it isn't necessary, and we'll see that the resistance to reprogramming is true even without DNA methylation. And there are a number of histone modifications, which I'll show only briefly. This is methylation, H3K for methylation. And if you look at the red and or green bars, with these various control regions, there's a big change in the first 24 hours as the histone becomes methylated. So now we come to transcriptional reprogramming. And the first general fact here is that this is not due to the so-called Yamanaka factors. You could say perhaps what happens is that the oocyte is packed full of the Yamanaka factors, and that explains how the reprogramming happens. But interestingly, the oocyte is largely insensitive to gene-specific factors. We've tested the Yamanaka factors, and if you overexpress the very effective master gene MyoD, it is almost completely ineffective in the context of an oocyte. So one of our conclusions I'll reach is that the oocyte does not use gene-specific transcription factors. Rather, it is a general reprogramming of polymerase that it seems to use. So I mentioned that as one point. The other relevant fact which I'll mention is that the oocyte, surprisingly, transcribes a whole lot of specialized somatic genes, like muscle genes and nerve genes. They are quite strongly transcribed in the female germline. So these are those 2 points. Gene-specific transcription factors do not seem to be effective in oocytes, and oocytes express this wide range of genes. Those are characteristics of this special cell. Now again, by looking for special components which are abundant in oocytes, one such comes up noticeably, and that's the so-called histone H3.3, which differs only slightly, 4 amino acids, from the core histone H3, but in other respects it's really very different, has a different chaperone, it's synthesized throughout all cell cycle phases, and its function always seems to be associated with high transcription. And you can see that this H3.3 histone moves into transplanted nuclei after the linker histone, about between 10 and 15 hours, quite a substantial occupation of transplanted nuclei by the histone H3.3. The anti-chaperone, HER-A, so chaperone, anti-chaperone, preventing it functioning will eliminate the effect, and so argue that it's a necessary step. Now, another interesting characteristic of the oocyte is that it is the oocyte that provides the polymerase II. You might have imagined that the incoming somatic nuclei, which have their own polymerase, would use that. In fact, that's not sufficient. And if you look for the normal reprogramming indicated by the transplanting nuclei becoming red, expressing these genes, that is eliminated by alpha-amanitin when you use alpha-amanitin-resistant nuclei. So these implanted nuclei will have functional alpha-amanitin. The oocyte, sorry about the spelling, the oocyte will have its alpha-amanitin eliminated, and that eliminates the transcriptional reprogramming. So we know that the oocyte has to provide this large abundance of its polymerase for this reprogramming to take place. The last thing I'll mention briefly is that actin polymerization takes place in the oocyte, in the transplanted nuclei, and my colleague Komiyamoto is analyzing this. It turns out to relate quite strongly to a component called wave 1, and this probably all connects with the transcriptional reprogramming. So to summarize the reprogramming side of activities, these are the main points. There is a global de-repression of somatic nuclei, and notably the oocyte linker histone is the first important step. Then there is change of histone modifications and some degree of DNA demethylation, and importantly, a really intense transcriptional activation involving a number of these components which we have mentioned. So this is a kind of time scale of this reprogramming which would be very hard to achieve if you have a low efficiency reprogramming system, and that's generally true of the IPS system. So here we are with on the time scale, relatively fast at 17 degrees, histone linker, histone transcription, methylation of that histone, special actin polymerization, and then the large amount of active transcription. So that is what I have to say about the reprogramming event. So how it is that a specialized nucleus can be reprogrammed, and again, to make the point that these somatic cells are very stable in their own cytoplasm, but once you change that, then they seem to be surprisingly sensitive to these conditions in the egg and being essentially returned to the state of the nucleus of the female germline. So now we come to the second part of the talk on resistance, and in my view this is perhaps one of the most interesting aspects of the whole field. That's because you may know that when you use the IPS system with transcription factor overexpression, there is an enormous amount of resistance to the switch. So even if you express the relevant factors strongly, the success rate of generating embryonic stem cells from somatic cells is actually very low. It's typically 1 in 1,000 or so, and by special methods it can be improved, but there's an enormously strong resistance to reprogramming, and the question is, what is that, and how might this relate to the stabilization of cell differentiation? Now, this is an old experiment, and I'm starting with experiments which relate to the eggs, and egg, again, I say, means these second meiotic metaphase recipient cells. And in these experiments, whether you use xenopus nuclear transplants which reach a tadpole stage, or you use mammalian nuclear transfers that reach birth, the success rate of nuclear transfer goes right down with time, with the more differentiated state. So as donor cells differentiate, the ability to reprogram goes rather dramatically down, as we mentioned at the beginning of the talk. Another aspect of this, which really shows a similar thing, in fact it shows it slightly more because it shows a kind of memory of a state. This is again an experiment where you transplant a muscle cell nucleus to an egg, grow the egg to an embryo, blastula, and then remove the middle part of the embryo, which will form muscle. And we're talking here of a muscle cell nucleus into an egg, making the embryo, take away the muscle lineage, but leave the neuroectoderm, nerve and skin derivatives, and the endoderm. And what you find surprisingly is that the neuroectoderm cells have a high overexpression of muscle genes, as do the endoderm genes, in spite of the fact that this nucleus, which was transplanted, has undergone no transcription for those 12 or so divisions. And when it starts up transcription again, with a sort of 50% success rate, it remembers where it came from. And that, if you make a guess, as we did, that this might have to do with the histone H3.3, you can overload the egg with a mutant form of this special histone as a competitor, and that eliminates the function of histone H3.3, and you then essentially eliminate the memory of this muscle existence of that nucleus. So it argues that this epigenetic memory can be explained by the fact that the histone H3.3 promotes continuing transcription of active genes. So when you transplant a nucleus with an active muscle gene, the high content of histone H3.3 in the egg makes that muscle gene transcription continue. Of course, it doesn't matter in normal life, because when a sperm enters the egg, it doesn't have any memory there, and therefore the H3.3 doesn't promote any kind of inappropriate memory. And it is true that the egg cytoplasm reverses this transcription with about a 50% efficiency. So in some cases you get completely normal development and there's no epigenetic memory, but in other cases this interferes with the success of the oocyte in reprogramming transplanted nuclei. Well, now I switch to try and analyze the mechanism of this resistance to reprogramming, and I now have to turn again to the oocyte, the first meiotic prophase cells, because these are so convenient for analyzing mechanism. And I can say something about the inactive X chromosome of female mammals. This is a very strongly repressed situation in which, as you know, the genes on the inactive X are switched off very completely, and they are very resistant to reprogramming. And this is shown, for example, here. We're looking at the OCT4 pluripotent gene transcripts, and if you look at mouse embryonic fibroblasts on the inactive X, there is no significant reprogramming. There's there is no apparent reprogramming whatever of the OCT4 in these mature adult mouse embryonic fibroblasts. The important point is that if you look at the inactive X on the so-called epiblast, which are a more embryonic stage, then you do get reprogramming quite substantially. The OCT4 is turned on because these more embryonic type of cells, nevertheless, with the inactive X, are more readily reprogrammed than the adult, more differentiated cells. And that gives an opportunity to find out what the difference is. And just to summarize a lot of work by my colleague Vincent Pask, this resistance turns out not to be explained by DNA methylation and not to be explained by the H3K27 methylation mark, experiments which show that. However, they are explained to a large extent by the histone variant macro H2A. The core histone looks like this, and the variant histone has a long extension here and is really a completely different molecule. It's a vertebrate-specific variant, and it's very characteristic of heterochromatin. So it looks as though this macro H2A is one component of the resistance to reprogramming. Example you can see in the non-reversible kind of myosin fibroblast, there is this mark, and in the epiblast type, this macro H2A is not there. So in conclusion, on this part, the macro H2A, by the way, can be knocked down, it's a good experiment, by inhibitory RNA, and when you do knock it down, you actually induce OCT4 and SOX2 genes in these adult MEF inactive X cells. So it says that macro H2A is one component which explains the resistance to reprogramming. So that's the conclusion. Macro H2A marks differentiation and acts as part of the resistance to reprogramming, and hence is a contributor to the stabilization of a specialized state. So now how can we progress from this? And we don't know the answer, but we're beginning to make some inroads into how one can find out what the whole explanation for resistance is. And the way one does this is to deplete components of chromatin, by which I mean isolated nuclei, and ask what it is you have to remove to get rid of the resistance. And the first guess that one might well make is that the non-coding RNA could be important because it's generally speaking quite inhibitory. And so the experiment is to remove that all non-coding RNA from a suspension of nuclei and then transplant the nuclei into the living oocytes where, as I've mentioned, they undergo recurrent reinitiation of transcription. And this can be done by using a mixture of ribonucleases to eliminate the RNA from donor nuclei. And this is after RNase treatment. And I should point out that this is a logarithmic scale. So we've reduced the RNA by about 5 orders of magnitude, leaving virtually none left in the nuclei. And of course, you then need the biological test of what has that done. But before doing that, we found that you can add RNA back again to these RNA-depleted nuclei, and that now persists. It really says that you have successfully removed the RNAs. Otherwise, of course, the experiment couldn't possibly work. So you have RNA-depleted nuclei which are able to be tested. And when you do this, to my surprise, it makes no difference to the reprogramming. So whether you're looking at OCT4 or SOX2, there is no difference between the normal nuclei and the RNA-depleted nuclei. They each show this almost identical increase in the accumulation of transcripts of these genes for OCT4 or for SOX2. Now that's very convenient because it seems to say that we don't have to struggle with trying to find out what kind of non-coding RNA might be responsible for the resistance to reprogramming. It looks like it simply doesn't play a part at all. So that leaves finally the question of proteins which may be on these nuclei, and one can deplete proteins from nuclei and chromatin by using high concentrations of salt and triton. But that gives you a very special problem, because when you do that, the nuclei swell up because the DNA is now more able to be released, and it forms a sticky gel which makes them completely impossible to transplant into an oocyte. We eventually found a way of encaging these nuclei, putting them in a kind of permeable mesh which fortunately stops the DNA swelling, and so you have then a preparation of these protein-depleted nuclei which can be transplanted and so tested for transcription and hence resistance to reprogramming. And this just shows that the high salt and triton does in fact remove a very large number of proteins. And when you do this, you can, looking at this part of the picture, the chromosomal proteins after salt and triton treatment are very largely eliminated. So there are a whole range of candidate proteins that might be expected to have some role in repressing transcription. H2A is a control, but in the others you go down to almost nothing left at the higher salt concentrations. And what the top of the picture shows is that when you're examining the gene SOX2, ES cells, of course, express this gene very strongly when they're transplanted to oocytes. But C2C12 cells, for example, are highly resistant to reprogramming of this gene. And to my surprise, when you remove all these proteins, it makes no difference at all. The C2C12 nuclei still show almost exactly the same resistance, even though nearly all the most obvious chromosomal proteins have gone. I should say we know that this resistance is not to do with DNA methylation, and that's rather easily shown by removing all the proteins from chromatin and transplanting DNA when there is no resistance. So this is, we don't know the answer to this, but it does imply that there is some kind of tightly bound, presumably protein, in the chromatin resistant to salt and triton treatment, which is, at least in this case, is responsible for the resistance. And it'll be interesting, of course, in future to see what that might turn out to be, as reflecting an explanation for the stability of the differentiated state. So in summary, for the resistance increases as cells differentiate. The inactive X resistance is explained in part by macro H2A, and some genes resist reprogramming by tight binding chromosomal proteins. Now this sort of summary diagram just puts the nuclear transfer work into perspective in respect of other kinds of reprogramming work. Now I've talked about transplanting nuclei like adult nuclei either into eggs when they can develop into these different derivatives, or into oocytes when they become, the pluripotency genes are reprogrammed. This really sets everything right back to the beginning of life, and you would have to go from the oocyte to egg to then let it progress along these normal differentiation routes. The Yamanaka IPS route, of course, goes from adult directly to embryonic stem cells, and hence to a range of these different cell types. And transcription factor overexpression, for example, using blood factors, particularly work of Thomas Graf, shows you can switch from a macrophage to a lymphocyte within that lineage by overexpressing the relevant transcription factors there. These factors would be hematopoietic factors, and they will go from one kind of blood to another, but they are unable to switch, say, blood to anything quite different. And then there's the pancreas work of Doug Melton, where they used pancreas-specific factors and were able to switch from exocrine to endocrine, again, just at that last stage of the lineage diversion. So the general conclusion would be that the further back in development you go, the more widely and effective are the reprogramming components of these cells. And as they undergo specialization and reach a stable state, they lose most of the reprogramming factors, which, however, are operative on the terminal lineages within the direction that they are intended to go to. So finally, here are the conclusions from what I've been talking about. First point, I'll emphasize again that the differentiation of whole cells is in fact very stable. We mentioned single cell transplants, and incidentally, some whole cells are determined amazingly early in development, like the the endoderm cells are already committed, irreversibly determined at the blastular stage. Then we talked about how nuclear transfer destabilizes cell differentiation and mentioned that the adult gene expression is reversed back to that of an embryo using eggs or oocytes. Somatic nuclei are reprogrammed mechanistically by chromatin decondensation, and we know a factor involved there, and by the general, not gene-specific transcription factors. So it's a combination of decondensing, opening the whole chromatin up, so access to genes is made very easy, and then a high concentration of general transcription factors that seems to account for the reprogramming. And then I mentioned that resistance to reprogramming, which I see as being equivalent to the stability of differentiation, which is really the topic of the talk, and that in some cases DNA methylation does help that, but there are many other mechanisms that also seem to achieve this stabilization, and we'll be interested to know what these tight binding chromosomal proteins might be that achieve that. My colleagues who've been contributing this work, Jerome Julien on the B4 histone, Kei Miyamoto, particularly the polymerized actin, Vincent Pasque on the inactive X, Richard Harris-Stott has done a lot of very careful quantitation. We try to measure everything in terms of the absolute number of transcripts per gene per time, and Kazumurata did the work on the histone modifications. Thank you.
Time for questions?
Yes, if you think there is, right, we'll see what we can do about that. Does it work?
That was a wonderful talk. I have two questions. The first has to do with the actin polymerization.
Yes.
Could you tell us more what you think that that's doing for reprogramming? And secondly, I know that many of the ATP-dependent remodeling factors, like switch-SNF type factors, either contain actin or actin-related proteins, and I'm wondering if you know if that family of chromatin remodelers is also involved in reprogramming.
Right, so this is protein remodelers, do you say, or chromatin
chromatin remodelers,
yes,
like switch-SNF type family
yes,
remodelers.
Well, I completely agree, and I suppose they must be. The problem is that we spend a lot of time trying to test that, and just to illustrate the difficulty, most of the protein remodeling complexes consist of about 20 different proteins, and the problem is to eliminate them from the oocyte, and we've tried, with a lot of help from Gerald Crabtree, by the way, to overexpress dominant negatives in order to get them incorporated into the oocyte maternal content of reprogramming complexes and hence inactivate them. And it's an open question as to how rapidly the exchange rate is within those 20 reprogramming molecules. And so far we just have not succeeded in being able to inactivate those complexes, but you're probably right that they're involved.
My second question has to do with that last experiment where you depleted proteins generally. Could it be that you're not only stripping the negative factors, but maybe also losing some positive factor that would be required for reprogramming?
Yes, right. So it's a good way of turning the question around. Well, I think you're right that we could. The way we're sort of going about it is to ask how you remove the resistance. So the expectation of that experiment is that as you take things away, you get an improved transcriptional activation, which in some cases you can. So if there were factors that, like you mentioned, were needed positively, then to remove those would make things get worse. So we rather expect that with that design of experiment, as you eventually remove most things, you should actually improve the results, and therefore it might not show up, the molecules that you mentioned, but you are right that they might exist.
It was a very nice talk, but I'd like to add a little bit more on the confusion side of really understanding what is going on. There was a paper published about half a year ago by you et al. In Nature, in which they showed that by simply overexpressing a neuron-specific microRNA in a fibroblast, you can convert some of these fibroblasts directly into neurons, which gives about a 5% yield. However, if you add then, in addition, a neuron-specific BHLH transcription factor, you can get that yield up to about 50%. Now the question to you, of course, is, what do you know about the microRNAs in your system, and most importantly, about the molecule, the enzyme that is making microRNAs, dicer-1, that it should still be there and should actually replenish your specific microRNAs in your oocytes?
Yes, well, I see your point, but there are microRNAs there, and it's an important question of whether they can be removed. And they would be a contribution from the oocyte, as you indicate, and that we have not tested. It's an interesting idea. All we've done is to say that such RNAs are not contributed by the donor nuclei. So I think it's a very good question, and it will. Actually, the range of microRNAs in the oocyte is very limited, and so it should be possible to outcompete the relevant one, which I think is called MIR 427, as far as I remember. So it's a good point, and it's well worth pursuing.
Eric? John, because MyoD can convert so many cells so efficiently to muscle, do you think muscle has a lower barrier for reprogramming than other cell types? Or do you think there's something really unique about MyoD that enables it to do that?
Yes. The experiment that I only mentioned in passing was the massive overexpression of MyoD in the oocyte, which has no effect of inducing muscle gene expression. That sort of argues that, I think what it really says is that that whole kind of overexpression of transcription factors is not what's going on in the oocyte, but your point is, could muscle be particularly easily reprogrammed, I think is what you're saying. It doesn't actually look very much like it. I mean, we compare this to neural genes and other kind of ones, and at least in the system we're using, it doesn't seem to be particularly more easily reprogrammed than others. But this is only one kind of test, and it may be that in the developing embryo there may be such an effect. That would be an interesting point if it's true.
I had a question about your macro H2A,
Yes.
which when you deplete, it favors reprogramming. I wonder a sense of how much it favors, and would it also favor reprogramming with the Yamanaka factors?
Yes. That has, we haven't done it in combination with Yamanaka factors. That would be an interesting thing to do. In many of these things, one would, answer your question a little bit indirectly. An obvious question which you might have said is, why not use B4 histone, linker histone, to pre-prepare donor nuclei for reprogramming, and so you would expect to treat the donor cells with this, and you might expect to see some difference if our ideas are correct. It turns out that experiment doesn't work because cells particularly dislike having an abnormal concentration of histone, so they essentially die. And there may be some very special concentration, which is exactly right. So those experiments are good ones, where you try to treat donor cells and see if you can actually improve the success rate. Ideally, you would be doing that in the egg system, where you transplant into an egg and get development. But it's a more technically difficult experiment, and I rather hope eventually these will work, but it's an obviously good question.
John, I wonder, what do you think the role of actin in reprogramming is? So it might be a possibility that it does relate to the chromatin remodeling, but is there another possibility to explain why actin is so important for remodeling,
why actin
for
is
reprogramming?
So important? Yes, why is it, you mean, essentially?
Why? And also I wonder whether if you eliminate actin, does reprogramming goes much less efficient or entirely stops? So how strong the effect is?
Well, we only know so much about the actin. There's an interesting anecdote that it used to be well known that if you mentioned nuclear actin in any grant proposal or paper, it would immediately be rejected on the grounds that you're bound to be dealing with an artifact. So if you want to get a rejection, that's one way of doing it. So, but actually, we don't know why actin polymerization is important. It does, my colleague is working with this wave 1, which actually seems to be a very important part of it. But why it does so and how it activates RNA polymerase, we don't yet know. So it's an unexplored area, but and I can't answer your question properly.
So I have one question here. Since your reprogramming system doesn't seem to involve S phase, so do you think DNA demethylation will happen in your...
Since the reprogramming happens in
your nuclear transfer experiment, it does, the DNA do not replicate, right? So there's no S phase, there's no de novo DNA synthesis,
no
and therefore there will be no DNA demethylation happening.
Yes. So you're saying that there is no demethylation?
There will not be demethylation, DNA demethylation. So you mentioned that later on that you're going to discuss that.
How much DNA demethylation really happens? There are some promoters where that does happen, not terribly efficiently, but there's an awful lot of others in this system. There is DNA demethylation in oocytes, but it's rather slow. And in many other cases, we deal with genes where there simply is no methylation of promoters, and yet the resistance that we're talking about is very pronounced. So it looks, my feeling is that DNA methylation is a kind of final step in really switching genes off. If you absolutely want them out of the way, then that's a good way of making them particularly resistant. But all the other steps along the way help to switch genes off and stabilize differentiation, but methylation is perhaps the final one that's most effective.
Right. Okay, then thank you the speakers very much for a very interesting night.